Finding the Galactic Underworld. I'm here to answer one question for you. What does it mean to hunt for stellar corpses? Well, what is the Galactic Underworld? When massive stars die, they end their lives in these huge explosions we call supernovae. Supernovae are so powerful that they can outshine an entire galaxy while they're active. But this explosion leaves behind a remnant, a core of now dead star, which is a neutron star or a black hole. However, as implied by their name, black holes are black, and as are most neutron stars. This means that we don't know where most of them are because we can't look up into the night sky and see them. But finding them is important because they can teach us about gravity, about dark matter, about the history of the galaxy, and much, much more. So, where are they? If we aren't going to stumble upon neutron stars and black holes by accident, then we need to work out where we should be looking. To work out where we should be looking was one of the questions I answered in my PhD. And a place to start might be, well, we know where the stars are, and neutron stars and black holes are dead stars. Could they be in the same place? Here is the visible Milky Way, our galaxy. Usually we draw it top down, so you can see those gorgeous spiral arms, but here it's side on. Okay, and our axes are the physical scale of the Milky Way. This here is the bulge, and then this is the plane of the galaxy. And if we were to put our sun on this diagram, it would sit at about here. The intensity of the color tells you how many stars are at that place in the galaxy. And up until now, we've assumed that the black holes and the neutron stars are where the stars are. They follow this same distribution. However, it turns out that that's not true. So, where are they? Well, I told you before that black holes and neutron stars are formed from massive stars. It turns out that most massive stars formed long, long ago. So if you trace back through billions of years, our galaxy had a different shape. It was taller and more diamond-shaped, like this. To work out where the black holes and neutron stars are now, we then have to evolve this galaxy through to the present day. Now, this is a complex process, so just let me highlight one step. When black holes and neutron stars are born in supernovae, the explosion is so powerful that they blast the black hole or the neutron star across the galaxy. And if we trace these remnants through the gravitational potential of the galaxy and then integrate to the present day, we reach a much more puffed up distribution, like this one. And here it is side by side with the visible galaxy. We can see the shape of the galactic underworld is dramatically different to the shape of the visible galaxy. Perhaps the most striking feature is that the height of the galactic underworld is triple that of the visible galaxy. I can also tell you how far away we'd expect the nearest neutron star or black hole to be. On average, it should be about 60 to 70 light years. Now, that may not mean much to you because perhaps you don't deal with these units every day. So let me put that in context. Astronomers consider everything within about 300 to 500 light years our local neighborhood. The nearest black hole we've spotted is a colossal 1,500 light years away. So 60 to 70 light years is practically next door. We haven't spotted them. They're just lurking out there in the dark. Because supernova are such powerful explosions, 40% of neutron stars and about 2% of black holes get blasted out of the galaxy entirely. This means that neutron stars are black and black holes are leaving the galaxy, never to return. But what's more is that because there's nothing special about our galaxy, all the other galaxies in our universe are also doing this, which means that the Milky Way is being peppered with neutron stars and black holes from other galaxies. We truly have intergalactic visitors. We just can't see them. To wrap up, the galactic underworld is a population of black holes and neutron stars, which are the remnants of dead stars. These objects will help us answer questions troubling modern physics, but we need to find them first. Black holes and neutron stars are dark, so for the most part, we don't know where they are. And it turns out that they should be in a very different location to the stars we see in our night sky. Just to finish on an upbeat note, not that that wasn't very exciting, but it's a really exciting time to be a black hole or neutron star fan, because there are telescopes coming online this year which will allow us to find many more black holes and neutron stars than ever before. Thank you.
Hi everyone, my name is Muyang Li, currently a third-year PhD student at the University of Sydney. I would like to start by thanking the Royal Society of New South Wales for this great opportunity to give this talk titled Measuring and Correcting Instance-Dependent Error in Semi-Supervised Learning. So what is semi-supervised learning and why is it important? We all know AI models have achieved this great success these days, and behind this glaring success, it is really machine learning that plays a pivotal role. When we have a closer look of machine learning, the most fundamental and common learning paradigm is known as fully supervised learning. This is a learning process that involves teaching the model the mapping from an instance, which could be an image, a sentence, or any abstract object, to an associated label, which could be another image, sentence, tag, etc. However, label usually requires fine-grained human annotations, which could be very expensive and timely to obtain, whereas unlabeled data itself are much easier to obtain. So consider images or sentences. You could use a simple web crawling tool to obtain a large amount of images or text at nearly no cost. But to know what these images are or what this text describes are very expensive. Therefore, a very intuitive and simple alternative has been proposed, which is semi-supervised learning, with the idea of learning a mixture of labeled and unlabeled data. And usually, the quantity of the unlabeled data is much larger than the labeled data. Now we have a general idea of what semi-supervised learning is, but how exactly do we learn a model from the combination of labeled and unlabeled data? The answer is actually quite simple. First, remember we have some data that are fully labeled, so we start the learning process by asking the model to fit those labeled data. After that, once our model have gained some ability, it will be applied to the unlabeled data and make predictions to them. This is what we call pseudo-labels. Like in this image, our model have already seen some images of cat in the label data. So when it sees a similar image in the unlabeled data, it will predict with certain confidence that this is also a cat image. Finally, if the prediction confidence of unlabeled data exceeds the confidence threshold, then this particular data point will be added to the label training set with its predicted pseudo-label. And we repeat this three-step process over and over to gradually expand the size of the label training data until all data are utilized or our model converges. However, while this intuitive process might sound promising, one significant challenge remains because the model we obtain usually have prediction errors, not to mention a relative weak model at the beginning of the training loop, which we know most predictions are bound to be incorrect. Therefore, the confidence threshold is very crucial to the success of this process. Most of the existing threshold techniques either employ the uniform threshold for all data or use class-dependent threshold, but those methods neglect the individual level information, which is not very desirable. Another challenge is known as the confirmation bias or error accumulation. This means during the self-training process, when model makes a prediction, it does not have the self-awareness on whether this prediction is correct or not. If the prediction is not correct, and the model still adds this data to the training set, then this error will accumulate and the model will blindly become more and more confident to the error it makes, rejecting correct predictions for other similar data. So is there a solution that allows us to tackle those shortcomings? Our research attempt to address these problems by modeling how the model tends to make mistakes. We leverage the fact that our data have both labeled and unlabeled data. Suppose the model indeed makes mistakes on the unlabeled data and overfits to those errors, then this will also be reflected on the labeled data as well. Since we know the ground truth label for the labeled data, it is easy for us to observe for this particular data point how the model are likely to make mistakes. For example, we can see here for this dog image is a posterior probability looks like this, which suggests that there is a notable probability the model will misclassify this dog to a loaf of bread. If all the predictions are correct, we expect that this predictive vector to be one hot, meaning only the entry on the dog would be 1 and the rest class should be 0. However, when we observe a posterior probability looks like the example here, this can serve as an indicator to probe the error that this model makes on the unlabeled data. This error can be stored in a transition matrix, which represents how the model makes the mistake in a classification task. And how exactly do we utilize the transition matrix? Well, there are two main advantages. Knowing that the transition matrix essentially models how the noise are generated, if we can obtain an accurate estimation of the transition matrix, we can therefore invert the noise generation process, hence correcting the observed noisy predictions towards the underlying true label. For instance, if we know with a larger probability this image will be misclassified from a dog to a bread loaf, then when we calculate the loss, we take this into consideration and penalize the loss value attributes to the entry of the bread loaf. Secondly, knowing an instance transition matrix helps us understand how likely this instance is to be noisy. If the off-diagonal of the transition matrix for this specific data point is large, then we know with a larger probability this image is likely to have incorrect label, and the threshold will be adjusted accordingly. And that is how we model the instant-dependent error and correct them. By doing so, not only we make the pseudo-label threshold instant-dependent, we also make the model aware of the error it makes, hence mitigating the aforementioned challenges and making the semi-supervised learning method more robust and reliable. Lastly, I'd like to use this time to acknowledge my supervisors, Professor Tongnan Liu, who recommended me for applying this award, and our collaborators who have contributed so much for the success of this project. And this project cannot be done without their support. So thank you.
Hi, my name's Joel. I'm currently a third-year PhD student in the Microwave Silicon Photonics Laboratory at the University of Sydney under Professor Jak Kelly, and I'm going to talk about optimization of silicon photonic circuits via machine learning methods. Thank you. So I'll just go briefly into the motivation as to why silicon photonic circuits are important for future technologies. And then I'll go into an application of machine learning optimization that is currently in use in research, as well as a device design method that is optimized by machine learning. So photonics integrated on chip has transcended traditional electronic interconnects owing to its high bandwidth density, intrinsic low power consumption, and reduced signal propagation loss, making it an attractive platform for communications and signal processing. Fundamentally, photonics is about light-matter interaction. On-chip photonics leverages this by trapping and controlling the transport of light through optical waveguides. The silicon on insulator platform is a widely used material framework on which these devices are built and is compatible with mature semiconductor fabrication schemes. Since photonic processing operates at micron or nanometer scale wavelengths, photonic circuits can be significantly miniaturized compared to electronic counterparts, with advanced applications in data centers, communication networks, sensing, quantum control, and AI accelerators. As the applications of these material platform grow in complexity, there is an exponential increase in the number of photonic devices integrated on chip. As a result, the core challenge of photonics research is to maximize scalability of circuits by reducing the footprint of these devices while maintaining high fidelity and bandwidth light propagation. Current research advances are directed towards such challenges that arise when designing and controlling integrated components at the nanoscale, namely fabrication errors and environmental factors such as temperature, resulting in degraded light confinement or phase variations. Common devices, such as vertical grating couplers, are used to couple the light from a laser source onto the chip and power splitters, optical mirrors, wavelengths, and mode demultiplexers can be integrated together in a photonic circuit, as shown on the left, to manipulate the propagation of light for use in communication and information processing. Optimization methods are required to account for and enhance both the design of the devices themselves as well as the broader system-level control, subject to these experimental imperfections. So conventional optimization methods typically involve starting with an initial set of design parameters, measuring each of these to yield a response, then manually updating them based on some decision-making, which is often limited in complexity by the intuition of the designer. On the other hand, inverse design, a subset of machine learning, takes a more intuitive approach. It starts instead with the desired objective and predicts, using a generator, a complete set of design variables that approximates its response, then automatically refines these simultaneously based on the ground truth error, which is important when validation of these parameters is expensive to compute or measure. This approach can be applied at both the component and system level, for example, in use on optical phased arrays. So the objective of an optical phased array control is to achieve a coherent beam spot at a precise predefined location, used to transmit and receive information on a satellite, for example, or as a high-resolution sensor for location and mapping on a robot. Phase shifters are used to align the relative phases of the optical antenna such that the diffraction pattern in the far field corresponds to this. By virtue of the dense integration of phase shifters, thermal crosstalk can alter the relative channel phase along the transmission line, resulting in an incoherent beam pattern. Machine learning in this case can be used to account for this by starting with the objective, a desired diffraction pattern, and querying it to predict a set of control currents to the phase shifters, implicitly accounting for fabrication and thermal crosstalk effects. By increasing the number of data collected, the machine learning model can refine its current prediction, achieving strong beam coherence under the influence of these experimental factors. At the device level, conventional methods make use of long waveguide channels to slowly transform optical modes. Scalability, however, is an issue for multiple integrated devices for applications such as optical interconnects. In this case, a wavelength demultiplexer is used to guide incident light of multiple frequencies to different ports for use in communications and signal processing. Inverse design methods reduce the footprint of devices by orders of magnitude through unique construction of a design region comprised of spatially varying refractive index. The topology of this structure is specifically crafted to guide the light into its respective output port in an ultra-compact form factor. By backpropagating the desired field response into the structure, the device material distribution is trained and optimized, leading to non-intuitive performant designs, which is inconceivable under conventional methods. These machine learning assisted methods yield great promise in tackling future scaling limitations as the complexity of on-chip photonic systems grow. Thank you.
Hello everyone, I'm Tian, a PhD student at the University of New South Wales. Today I'm excited to share some of our research on using nanoparticles for drug delivery. Many research are focused on drug discovery for cancer. However, a common challenge is those drugs are frequently too hydrophobic, leading to limited or abandoned further development. Instead of discovering new drugs, our group focuses on improving the drug delivery within human body, which can potentially save those nearly abandoned drugs or improve the efficacy of existing drugs. Instead of just relying on the drug itself, drug delivery systems use well-compatible materials as drug carriers, acting like vehicles to transport drug passengers through the body until they reach the target site. Since the drugs are trapped inside the carrier, they can be protected from degradation. Moreover, the solubility of this formula is now dependent on the material of the carrier instead of the drug themselves. Furthermore, the carrier can be modified with targeting units, which can specifically recognize cancer cells or a desired organ to achieve targeted delivery. So, what can be used as drug carriers? One emerging material is nanoparticles, especially polymeric nanoparticles. A good example of natural polymer is protein, which is formed by the folding of polypeptide chains. But now, with the rapid development in polymer chemistry, researchers can make a variety of polymers in the lab using synthetic materials. These synthetic polymers can be engineered into nanoparticles with diverse structures and properties. So, every drug delivery system is designed for a specific organ. Perhaps the most difficult is the brain due to the existence of the blood-brain barrier. The blood-brain barrier is a highly selective and protective barrier which maintains the brain's stable environment. It selectively allows the essential nutrients such as glucose, amino acid, and oxygen to enter the brain and restricts the entry of harmful substances. But this highly selective permeability also presents great challenges for drug delivery to the brain, as most therapeutic drugs cannot cross the blood-brain barrier. So, nanoparticles as drug carriers offer a promising solution for drug delivery to the brain. One crucial aspect is the incorporation of brain targeting units that can be specifically recognized by the blood-brain barrier. Also, because the blood-brain barrier consists of a monolayer of endothelial cells connected by very tight junctions, the smaller nanoparticles are generally expected to cross more easily than larger ones. Therefore, inspired by natural self-folded proteins, our effort is to design self-folded small synthetic polymeric nanoparticles for brain drug delivery. In my PhD project, I have synthesized a polymer consists of 90 units of glucose and 30 units of tyrosine. The glucose enables brain targeting by interacting with glucose transporters overexpressed on the blood-brain barrier, while the hydrophobic tyrosine promotes the polymer chain folding and interaction with hydrophobic drugs. By simply mixing the polymer and drug in water, efficient drug encapsulation is achieved by forming small drug-encapsulated nanoparticles of around 10 nanometers in size. This approach serves several advantages, such as biocompatibility, straightforward drug encapsulation process, and small nanoparticles with brain targeting capabilities. Here is a comparison of toxicity between a very hydrophobic drug, represented in blue, and the drug-loaded nanoparticles shown in yellow on cancer cells at two different drug concentrations, while the control is where the cancer cells are left untreated. The results demonstrate significant reduction in cancer cell growth with our nanoparticles compared to the drug alone. This suggests that the nanoparticles help to deliver the drug to the cancer cells, whereas the drug itself, due to its hydrophobicity, is likely to precipitate and thus cannot be absorbed. This highlights the potential of nanoparticle-based approach as a more efficient and reliable strategy for cancer therapy. In the future, we aim to further optimize nanoparticle formulation to improve the drug loading efficiency and the stability. And also, we would like to explore the delivery mechanism of very small nanoparticles in cancer cells, paving the way for more personalized and targeted treatment options. Finally, I'd like to express my gratitude to my supervisor, Professor Martina Stenzel, for her support, and thank the Stenzel Group and my secondary supervisor, Juning Ho, also the School of Chemistry at UNSW. Thank you, everyone.
Hi, this is Dr. Deng from University of New South Wales. Currently, I'm an NHMRC Year 1 Fellow and also NSW Cancer ECR Fellow. My presentation topic is Advancements in CRISPR-based Diagnostics. As we know, in 2020, the Nobel Prize in Chemistry was awarded to two female scientists for the development of a method for gene editing. This method is CRISPR-Cas9-based gene editing technologies. As shown in the right figure, CRISPR-Cas9 technologies contains two parts. One part is the Cas9 protein. Another part is the guide RNA. After the Cas protein and the guide RNA form the complex, it will be applied for the recognition of the target gene. After it recognizes the target gene, it will trigger the cis cleavage functions for gene editing. These technologies can be successfully used for human gene editing. In further research, we further developed these technologies for diagnostics as shown in this schematic figure. After the target gene was being recognized by the Cas RNP, it will trigger the transcleavage of Cas RNP for the cleavage of the fluorescent reporter, which can be used for the biosensing applications. This CRISPR-Cas-based biosensing technologies shows very good sensitivity and also specificities. It can be used for the detection of DNA, RNA, protein, and also small molecules. However, if we only use CRISPR-Cas systems for detections, the sensitivity can only be visualized in picomolar level sensitivity, which was not sufficient for clinical applications. So, in current stage, most of the technologies they used pre-amplification technologies to combine with CRISPR-Cas technologies to visualize point-of-care detections. These pre-amplification technologies including PCR, RCA, RPA, or LAMP. However, if we use these pre-amplification technologies, it will cause potential amplicon contaminations, leads to false positive results, and it also sometimes requires the instrumentations. So in our research, we developed a CRISPR-Cas-based autocatalysis systems as shown in the left figure. One target DNA or RNA activates the CRISPR-Cas systems. It will transcleave the circular mediators we designed. After it linearized this circular mediator, it will become a fake target to activate a new Cas RNP to form these autocatalysis systems. It changes previous 1:1 ratio to one target to multiple Cas RNP systems. So these systems successfully realized PCR level sensitivity in 15 minutes at room temperature. So it do not require instrumentation and also trained procedures. We have successfully demonstrated these systems for early cancer diagnosis in animal models, and also we successfully used this one for bacterial detections and also virus detections such as COVID. So currently this technology has been published and it also been patent protected. Up to now, our technologies realized TRL5 pilot scale. We are aiming to next stage, TRL6 large scale, so we appreciate collaborations for the commercialization of these technologies. So finally, I would like to thank you my team and also thank you my collaborators and also funding support. If you are interested in our technologies, please feel free to contact us. Thank you.
Hi, I'm Dr. Jen Matthews, and I'm going to take you on a deep dive into coral nutrition. First, I'd like to acknowledge the traditional custodians of the land and sea country on which I conduct my research, and pay my respects to the elders past, present, and emerging. Coral reefs are foundational ecosystems with immense ecological, economic, and cultural value. Healthy reefs support about 7,000 marine species. They provide billions of dollars, millions of jobs, protect our coastlines, and are an important source of new medicines, for example, medicines for Alzheimer and cancer. In Australia, we often consider that the Great Barrier Reef is the main hub for corals. But New South Wales has some pretty impressive coral gardens that support local economies, including tourism and seafood industries, and contribute to the unique marine life New South Wales has to offer. But climate change is causing the catastrophic loss of corals in our lifetime and on our doorstep. The corals in coastal New South Wales are no exception. In 2016, about 60% of New South Wales corals experienced bleaching as a result of the abnormally high ocean temperatures. And these high temperatures are also bringing invading species that threaten the immense ecological and economic value the New South Wales marine life has to offer. And without help, coral reefs as we know them may not survive this century. I've dedicated my career to increasing our knowledge of what determines coral health and resilience, to better inform conservation strategies and help to safeguard our reefs and the services that they provide for future generations to enjoy. One important research avenue I explore is coral nutrition. Just like you and I, corals need nutrients to thrive and survive, and they get most of their nutrition from a plant, a single-cell algae that lives in their tissues. And like us, optimal nutrition is especially important when they are at their most vulnerable, for example, as babies, or when they're sick or stressed. And when the water gets too hot, the algae can leave the corals, and so you see right through the coral tissue to the white skeleton underneath, giving them a bleached appearance. Now, bleached corals are not dead, but they don't have their main food source, and if bleached for too long, they will starve. I investigate which nutrients differ between corals under different conditions, like bleached versus unbleached corals, to better understand how nutrition determines coral health, or how some coral species can adapt to new environments like those migrating down towards Sydney, and to develop new management strategies like diagnostic tools or nutrient supplements. And feeding corals when they're vulnerable could help them to survive stress, just as a multivitamin helps us when we're sick, or it could enhance their reproductive success, like how we take pregnancy supplements. And an example of my nutritional supplement in action is our coral baby food. Corals produce millions of tiny little babies in a magical event a few days after full moon in spring, but typically less than 1% survives. And this is mainly thought to be because they just don't have the energy or food to make it to adulthood. So I went to the Great Barrier Reef for this once-a-year event, and with my team, we collected millions of tiny babies directly from the reef. We fed them with different types of baby food, and after 6 months, we found that more than double the number of corals survive when they're fed versus those that weren't. So it works. And something else cool and unexpected we found was that feeding actually supported their swimming. I know you're always told to not eat before you swim, but it seems to work in corals' favour. And when you feed corals, they go from swimming slowly and steadily to doing little coral zoomies. They swim further and faster. And this is really important when we consider reef connectivity. For example, healthy reefs could help to restock nearby degraded reefs if the larvae can swim there effectively. And while ambitious and unlikely to cure bleaching, it's this kind of research and interventions that will help to buy time for reef until we get our act together on climate change. And I am optimistic. I believe that it is possible to protect our coral reefs. And while it's unlikely that they will look like our reefs of today, protecting the diversity of existing reefs through management strategies could help reefs to adapt to future climate conditions, whatever they may be. But the time to act is now. Let's safeguard our reefs through science, innovation, and collective action. Thank you.
Hi everyone, my name is Cynthia Turnbull and I'm a postdoctoral researcher at the John Curtin School of Medical Research within the Australian National University. Today I would like to talk to you about why I think some people develop severe disease while others don't, and also touch on to how we can use our knowledge of genetics to potentially modify the immune system and treat disease one protein at a time. I'll start by saying that the immune system is an intricate network of cells and proteins which seamlessly works together to fight off an array of different foreign invaders, whether they be microbes, toxins, or allergens. It does this with incredible potency, while also knowing not to hurt the body in the crossfire. Unfortunately, mutations or changes in our DNA can alter the normal functioning of the immune system and lead to many of the diseases that impact our society today. For example, we can have mutations that lead to a weaker immune system and the development of infection and cancer, but we can also have mutations that cause the opposite effect and the overactivation of immune cells which attack the body and drive autoimmunity and the development of autoimmune disease. Despite decades of research and the identification of many different mutations, there is still a major obstacle in taking what we know about the genetics of the immune system and translating it to the development of better therapies and the eradication of disease. This problem is the strange phenomenon of incomplete penetrance, which describes a situation where we have patients with severe disease and damaging mutations shown in black, but also an unexpectedly high percentage of individuals who carry the same mutation, or carriers, but have no symptoms or only mild disease. These individuals are shown in grey. This can be quite high, up to 40 to 50% for certain diseases, and is a major challenge for genetic counselling, drug design, and treatment. In fact, current immunotherapies which target immune cells and proteins have only 20 to 50% efficacy rates, which simply isn't good enough for the eradication of disease. So why do some people develop disease while others don't? There are a couple of different theories, but today I would like to talk to you about modifier proteins. These are special proteins in immune cells which can modify the effect of damaging mutations, either making them more harmful or harmless. Through my PhD, I identified a new modifier of the immune system, the Dectin-1 protein, and used it to explain a very confusing case of incomplete penetrance. I did this by studying a patient with a severe form of autoimmunity. He has a disease called IDEL, immune dysregulation with autoimmunity, immunodeficiency, and lymphoproliferation. I know that that's a lot of big immunology words, but what this means is that the patient had an expansion of overactive immune cells and features of autoimmunity, including type 1 diabetes, gut inflammation, and eczema, but he also had immunodeficiency and a high susceptibility for both bacterial and fungal infections. For example, he'd had a yeast infection, but also a chronic salmonella infection. And so, by sequencing the DNA of the patient and his two parents, we identified that he had inherited a mutation from his mum that was known to cause autoimmunity. But this was with incomplete penetrance. You can see here that despite carrying the same mutation as her son with very severe disease, the patient's mum was actually quite healthy. She'd only had one mild flare of autoimmunity, which was easily treated. Instead, we think that the patient's disease, or the severity of it, was being triggered by a second mutation in Dectin-1, which he had inherited from his dad. Dectin-1 had already been described as an important protein for fighting off different bacterial and fungal species which were causing infection in the patient, and so this could explain the immunodeficiency side of his disease. But through my PhD, I discovered that Dectin-1 could also regulate the immune system. Dectin-1 is able to do this by taking T cells and turning them into a special kind of T cell called a T regulatory cells. These Tregs are effectively the brakes of the immune system and stop overactive immune cells that can expand during infection, like what the patient had, from turning against the body and causing autoimmunity. And so, by identifying that Dectin-1 was a modifier in my patient, we were able to explain why he was so sick while his mum wasn't. Because of the Dectin-1 mutation, the patient was unable to make more Tregs and stop his overactive immune cells from causing autoimmunity, while the patient's mum, who had a working Dectin-1 modifier protein, was able to make more Tregs and put a brake on autoimmunity before it could spread across her body. And so my goal now is to identify more modifier proteins like Dectin-1 by working with patients with different forms of disease and using animal models to mimic human disease states. I hope that this will expand our knowledge on how the immune system is regulated, but also I'm so excited to continue this research because I believe that modifier proteins could be used as little remote controls for turning the immune system on and off, and an excellent way for us to treat disease. I'd like to finish by thanking everyone who's been involved with this project, including my supervisors, Carola and Pablo, and of course my patient and his family. Lastly, I would like to thank the Royal Society of New South Wales for the incredible honour to speak today, but also to be a recipient of my award. Thank you so much.